on Planetterrian Daily, episode 105. It's June 29th, 2026. Let's get into today's research and health developments. Plasma protein patterns now flag organ-specific risks for dementia, heart disease, and several cancers years ahead. Researchers analyzed roughly 7,000 proteins in plasma samples to build organ-specific proteomic clocks. The clocks tracked risk for all-cause mortality, cardiovascular events, dementia, and multiple cancers. Lifestyle factors such as physical inactivity, smoking, and heavy alcohol use shifted the clocks in measurable ways. This work replicates earlier proteomic aging signatures with an independent platform. A few episodes back, the show examined aging biomarkers. Today's study adds organ-specific resolution that clarifies which tissues drive individual risk trajectories. From molecular signatures of aging to practical interventions that can move those same risk trajectories, we turn to exercise. A 6-month randomized trial of more than 120 participants in their 70s compared high-intensity interval training with moderate and low-intensity exercise. Only the high-intensity group lost fat mass while maintaining lean muscle. Moderate and low-intensity programs produce smaller fat losses accompanied by muscle decline. The differential effect highlights intensity as a variable that protects sarcopenia risk during weight reduction. The result sharpens questions about intensity thresholds that protect against sarcopenia in older adults. While intensity protects physical resilience, another preserved capacity, language processing, remains active even when consciousness is absent. Patients under general anesthesia process nouns, verbs, and adjectives while hearing stories, with neural signals showing anticipation of upcoming words. Researchers recorded activity from implanted electrodes during routine surgical anesthesia. The findings indicate that language networks remain active even when consciousness is suppressed. The data challenge the assumption that unconscious states eliminate complex linguistic computation. Most people picture general anesthesia as a complete shutdown of brain activity. In reality, the brain continues sophisticated processing even while the person remains unresponsive. Right now, as you listen, your own language networks are predicting the next word in this sentence. Similar predictive signals appear in anesthetized patients hearing stories. The practical takeaway is that unconscious states are not uniform blanks. Future brain-computer interfaces may exploit these preserved pathways for communication when patients cannot move or speak. Now, shifting to a very different area of research. A modified large serine recombinase now directs precise integration of sizable DNA payloads at chosen genomic sites. The retargeting method avoids random insertion and reduces off-target events. Tests in human cells demonstrated one-step placement of multi-kilobase sequences. The tool expands options for stable gene addition beyond CRISPR cut-and-paste approaches. Precision genome tools often rely on accurate protein structure predictions. A new method now improves those predictions for flexible proteins. AlphaFold 3 typically outputs one dominant conformation even when proteins sample multiple states. Researchers at the Institute of Science and Technology Austria combined the model with experimental data to generate ensembles that match measured conditions. The method corrects for local structural changes that depend on environment or ligands. It improves accuracy for flexible regions without retraining the underlying network. Follow-up work will test whether the same guidance improves predictions for membrane proteins and large complexes. While structural tools advance, immune cell lipid handling offers a different window into disease progression. Brain tissue from patients with aggressive multiple sclerosis contained abundant foamy macrophages filled with fat droplets after they engulfed damaged myelin. These lipid-overloaded cells switched from repair functions to sustaining inflammation. The observation links myelin debris clearance failure to faster disease worsening. Researchers are now examining whether clearing the lipid load can restore protective activity. from cellular mechanisms inside the body to mechanisms that shape entire ecosystems outside it. Grasslands covering 40% of land surface were studied to test how the sequence of extinctions affects ecosystem resilience. Removing certain species first reduced resistance to drought and nutrient shifts more than random loss. The work shows that functional traits of the first species to disappear determine how much productivity and carbon storage remain. Models that ignore loss order therefore underestimate future risks to food production and biodiversity. 2-year trial findings add another data point on supplements and brain health. Fish oil capsules raised brain omega-3 levels yet produced no measurable change in memory, cognition, or Alzheimer's-related imaging markers. The study followed participants for 2 years and directly compared supplemented and control groups. Results shift focus from isolated supplements toward broader dietary patterns that deliver the same fatty acids. The null finding aligns with earlier trials that also failed to show prevention effects in cognitively healthy adults. Beneficial microbes stimulated lignin production in plant roots rather than blocking salt uptake. Greenhouse and field trials showed improved growth and yield on saline soils after bacterial inoculation. The mechanism gives crops a structural buffer against osmotic stress. The approach offers a biological alternative to chemical amendments for reclaiming salt-affected farmland. Long-term exposure data strengthen links between a common pesticide and neurodegenerative risk. UCLA researchers linked residential proximity to chlorpyrifos with more than twice the likelihood of developing Parkinson's disease. Laboratory experiments showed the compound damages dopamine neurons and impairs clearance of toxic proteins. The population-level association strengthens the case for examining pesticide residues in Parkinson's epidemiology. A living relative of the earliest land plants revealed a dynamic sunscreen mechanism that adjusts to ultraviolet exposure. The system likely protected the first terrestrial flora more than 500 million years ago. The study traces how this biochemical toolkit was retained and modified in modern plant lineages. University of New Hampshire analysis of solar events showed measurable drops in rain and snow in the hours and days afterward. Stronger storms produced larger reductions. The link operates through atmospheric ionization changes that alter cloud microphysics. The pattern offers a new variable for short-term weather and climate modeling. The James Webb Space Telescope identified water vapor, methane, carbon dioxide, ammonia, and previously unconfirmed salt clouds in the atmosphere of a world 57 light-years away. The salt aerosols help explain the planet's persistent pink hue. Spectral data now allow direct comparison of cloud chemistry across the growing sample of directly characterized exoplanets. An associated set of fossil vertebrae from Otodus megalodon was rediscovered after missing since the 1980s. Danish and international researchers re-examined the specimen. Earlier size estimates reaching approximately 80 feet received support from the vertebrae measurements. Bone proportions also suggest lifestyle details about swimming efficiency and prey size. The find closes a long-standing gap in the megalodon fossil record. 2 intentionally mummified dogs from southern Peru date to roughly 1,100 years ago. One young animal was placed on a woven mat in a small pit, positioned as if sleeping. The burials predate the later use of dogs as status symbols in Andean societies. The arid environment preserves soft tissue and wrapping materials that reveal deliberate funerary treatment. The finds expand the timeline for companion animal relationships in pre-Inca cultures. Next time, keep an eye on follow-up work testing whether guidance from experimental data improves AlphaFold predictions for membrane proteins. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time, and if you'd rather watch than listen, find us on YouTube at NARA Network, links in the show notes. And before you go, this show is part of the NARA Network, a family of daily podcasts on tech, science, and markets, each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. SpaceX Daily, the daily companion for following SpaceX as a public company, from Starship to the stock ticker. Find every show free at narrownetwork.com. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.